Chemistry of life. Biological macromolecules. Carbon can combine with other elements, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, to make the large, complex, varied molecules that are found in all of the diverse life forms on Earth. The molecules of life, called biomolecules or macromolecules or biological macromolecules, fall into the following categories: carbohydrates, nucleic acids, proteins, and lipids. Carbohydrates, proteins, and nucleic acids can be very large, and so they are called macromolecules, where macro means big. The International Union of Pure and Applied Chemistry defines a macromolecule as a molecule of high relative molecular mass, the structure of which essentially comprises the multiple repetition of units derived, actually or conceptually, from molecules of low relative molecular mass. Lipids are smaller, and so they're not always called macromolecules. They don't quite follow the same rules as the other large biological molecules, so they will be covered last. IUPAC defines a monomer molecule as a molecule which can undergo polymerization, thereby contributing constitutional units to the essential structure of a macromolecule. IUPAC defines a polymer as a substance composed of macromolecules, very large molecules with molecular weights ranging from a few thousand to as high as millions of grams per mole. Carbohydrates, proteins, and nucleic acids are polymers. Polymers are built by covalently bonding smaller building blocks called monomers. Mono means one, mer means unit. Carbohydrates, proteins, and nucleic acids are each built by covalently linking many of their monomers. Many monomers linked together form polymers. Macromolecules are polymers. Poly means many. Polymers can get very large, made up of thousands of monomers and billions of atoms. This table identifies the type of monomers used to build carbohydrates, proteins, and nucleic acids. So carbohydrates are made of simple sugars. Proteins or polypeptides are made of amino acids, and nucleic acids are made of nucleotides. Peptide refers to proteins. A peptide or polypeptide is a chain of amino acids. Saccharide refers to carbohydrates, as in polysaccharides and monosaccharides. There is a limited number of each type of monomer. There are 3 common monosaccharides and many derivatives that build every carbohydrate. There are only 20 different amino acids and a few derivatives that build every protein. There are 5 common nucleotides that build every nucleic acid. And there are a limited number of lipid structures. Polymers can also be classified by how many monomers are combined in their formation. If 2 monomers combine with a new covalent bond, a dimer is formed. If another monomer is added, a trimer is formed, and addition of another forms a tetramer. More specific vocabulary uses appropriate suffixes. A molecule with 3 amino acids is a tripeptide. A molecule with 2 monosaccharides is a disaccharide. A molecule with 4 nucleotides is a tetranucleotide. A molecule with 6 to 20 nucleotides is an oligonucleotide. A molecule with more than 20 amino acids is a polypeptide. All organisms share the same limited number of monomers, yet each organism is unique based on the unique arrangements of their monomers in polymers. Changing the sequence of monomers in the chain changes the shape and properties of the chain and the function of the polymer. An immense variety of polymers can be built from a small set of monomers with limited modifications. Polymers are formed through a process called dehydration synthesis. 2 monomers combine, a water molecule is removed, and a new molecule, a polymer, is created. This process is illustrated on the next slide. Dehydration means removing a water molecule. Synthesis means the combining of separate parts to make a new whole. Here's molecule 1, here's molecule 2. A molecule of water is removed and a new molecule is formed. New molecules can also be formed by breaking existing molecules apart, allowing the products to react with other molecules. The breaking apart process is called hydrolysis. A water molecule is added to a molecule that causes both molecules to split apart. The hydrogen and OH groups from the water will then combine with each product, as illustrated on the next slide. Adding a water molecule splits the original molecule into two molecules. Here is a summary showing dehydration synthesis. You have two monomers becoming a polymer and water. And here we have hydrolysis, which is a polymer plus water becoming two monomers. Each class of biological macromolecules can also be described as being built from functional groups that modify a carbon-containing backbone. Carbon-containing backbones are a string of mostly carbon atoms with some oxygen, nitrogen, and phosphorus atoms. Functional groups are subunits of only a single atom or few atoms present in a series of compounds that impart particular chemical and physical properties when attached to other molecules. The key functional groups found in the four classes of biomolecules are hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl.